Lessons from pathological analysis of recurrent early esophageal squamous cell neoplasia after complete endoscopic radiofrequency ablation. Introduction. Endoscopic radiofrequency ablation, RFA, is increasingly being used to eradicate Barrett's neoplasia, as well as the flat type of early esophageal squamous cell neoplasia, ESCN. Previous studies have shown good short-term outcomes of RFA in treating early ESCN, with a complete response rate ranging from 84% to 97% at 1 year, and a low adverse event rate. The ablation effect of RFA on the human esophageal wall is uniform and superficial, with a maximal ablation depth to the level of the muscularis mucosae layer, approximately 1,000 micrometers. Compared with other endoscopic treatment modalities such as argon plasma coagulation, APC, photodynamic therapy, and endoscopic submucosal dissection, ESD, RFA therapy has several advantages over other ablative therapies, including uniform ablation, controlled depth, and the advantages over ESD, such as lower stricture or perforation risk. However, long-term follow-up studies are still lacking. In addition, the patterns of recurrent and persistent ESCN after initial successful RFA have not been reported. Therefore, studies to elucidate whether RFA therapy is associated with a high recurrence rate, as with other tissue-destructive therapies, are warranted. The major concern with RFA is the lack of post-treatment specimens to evaluate the curability of the whole lesion. Moreover, extension of neoplasia along mucosal surfaces into gland orifices and ducts of esophageal submucosal glands has previously been reported in ESCN. However, whether these involved ducts beyond the reach of ablation will create a sheltered niche or even lead to a buried squamous cancer has not been elucidated. Therefore, this study aimed to evaluate the treatment outcomes, identify the risk factors for tumor recurrence, and determine whether buried ESCN exists in recurrent or persistent ESCN after RFA therapy. Background. Endoscopic radiofrequency ablation, RFA, is a treatment option for early esophageal squamous cell neoplasia, ESCN. However, long-term follow-up studies are lacking. The risks of local recurrence and buried cancer are also uncertain. Methods. Patients with flat-type ESCN who were treated with balloon-type plus or minus focal-type RFA were consecutively enrolled. Follow-up endoscopy was performed at 1, 3, and 6 months, and then every 6 months thereafter. Endoscopic resection was performed for persistent and recurrent ESCN, and the histopathology of resected specimens was assessed. Results. A total of 35 patients were treated with RFA, of whom 30, 86%, achieved a complete response, 3 were lost to follow-up, and 5, 14%, developed post-RFA stenosis. 2 patients had persistent ESCN and received further endoscopic resection, in which the resected specimens all revealed superficial submucosal invasive cancer. 6 of the 30 patients with successful RFA, 20%, developed a total of 7 episodes of local recurrence, mean size 1.4 centimeters, during the follow-up period, mean 40.1 months, all of which were successfully resected endoscopically without adverse events. Histological analysis of the resected specimens revealed that 6, 86%, had esophageal glandular ductal involvement, all of which extended deeper than the muscularis mucosae layer. Immunohistochemistry staining for P53 and Ki67 suggested a clonal relationship between the ductal involvement and epithelial cells. None of the tumors extended out of the ductal structures. No cases of cancer buried beneath the normal neosquamous epithelium were found. Conclusions. Because ductal involvement is not uncommon and may be related to recurrence, the use of RFA should be conservative and may not be the preferred primary treatment for early ESCN.